actually allow these bacteria to actually cause infection. We'll talk about that today. We'll talk about a little bit more on Wednesday. And then that'll end with Carol for the second exam, which will be next Monday. We'll talk about the second exam here. We'll work on Wednesday. Not a long lecture. I don't anticipate us being here until 10:15 today. I hope not. Because I like to get long ones. So we'll talk about this and then we'll do a street plate, which will be real easy to do as well. We've all had micro, so we've done a street plate. We've done a street plate before, right? We've done micro. Okay, good. Whew. We did a street plate today. What I did is I mixed E. coli and serratia marcescens. E. coli is like a tan color. Serratia, if you grow it at room temperature, makes this pretty like pinkish rose color kind of pigment. So you can actually see when you separate the individual colonies because some will be pink, some will be tan. It's really easy to distinguish the two. So we'll do that here today as well. Alright. So a little bit about terminology. So the difference between pathogenicity and virulence. So when you're talking about pathogenicity, that's the ability of a microorganism to be a pathogen. So can this microorganism cause infection or not? Can it cause pathogenesis? So the ability to cause disease. When you talk about virulence, it's usually how sick the patient gets. If you're talking about virulence, you already know you're talking about a pathogen. It's just how sick that pathogen is going to make the patient. Now, in the past, in Gen Micro, you should have this too. The difference between infectious dose and lethal dose. What's an ID 50? Do you remember what infectious dose 50 is?
To get 50 people sick, 50% of people sick.
Right. So you have, say you have 10 mice, and you infect those 10 mice with a dose of some kind of pathogen. It's the dosage that makes 50% of those mice sick, so 5 out of 10. That's it. You run a bunch of different trials, groups of 10 mice at a time. Each group of 10 mice gets a different dose. The dose of which 50% of those mice are sick is your ID 50. What about your LD 50, your lethal dose 50? That's a little bit easier to wrap your head around. What about LD 50? What's that? Yeah, what dosage it kills half of them. That's it. If you look at that graph over there, which color line would you be the most concerned about? Red? Is that blue? Red, blue, or black?
Red.
Yeah. Why red?
Because it's the smallest amount of it.
Right. It takes less to kill 50%. If you're looking at things like infectious dose, some of the dosages for some of these pathogens, like say measles or rabies. Rabies is even a better example. So infectious dose is like one rabies virus. So it's really, really low. And there's some other examples of that too. We'll talk about as we go through. Virulence factors or components, specifically bacterial cells are going to make that's going to allow them to be virulent. Things like it might be a bacterial cell that makes a hemolysis and lyse red blood cells. That will also be a virulence factor. Depending on how much of a rigorous you are with these definitions, virulence factors are supposed to describe things of the protein nature that are made by bacterial cells that allow it to be virulent. Like your hemolysis, leukocidins, collagenases, hydroxylases, things that are going to be made by the bacterial cell that allow it to establish an infection and be pathogenic somehow. However, there are other things too, like bacterial capsule. Now, it's not like some kind of protein that's secreted by the bacterial cell, but it would probably be virulent if it didn't have that capsule, or it would be a lot less virulent. So there's a little bit of nuance to what a virulence factor is. But ultimately, it's something that's made by microorganisms that allows them to cause virulence. Pathogenic bacteria are going to have a couple qualities. We'll talk a little bit more about these as we go throughout these slides today. But invasiveness, so that's the ability to invade host tissue. When they invade, they have to have the ability to colonize. The number one thing that needs to happen for, say, a bacterium to establish an infection is it has to attach to your cells somehow. Whether that's respiratory tract, or your skin, or your gastrointestinal tract. There has to be some mechanism where that virus, bacterium, protozoan, whatever, attaches to you. If it doesn't attach, it can't establish an infection. This is I gave an example of E. coli and here causing UTIs, right? Maybe already. So this is kind of goes back to that example. If you have somebody come in to the hospital and you collect your urine specimen, there's E. coli in it. That doesn't necessarily mean they have a urinary tract infection. If they have E. coli in their urine, but they don't have any symptoms of urinary tract infection, they might just have E. coli in their urine. It's not doing anything. Sometimes you get E. coli in your urine, but that E. coli doesn't make the villi that are needed to attach to the urogenital tract, the urinary tract. So if you have E. coli in your urinary tract, they can't attach to your urinary tract. Eventually, it's going to get out. It's just going to flush out. So you don't need to treat those kinds of infections. So why can't they colonize establish infection? Because it can't attach. So ability to colonize, ability to produce extracellular substances that facilitate invasion. Oftentimes, there's many examples where these proteins are called invasins. Different bacteria will make different virulence factors that allow it to get into your tissue. So some bacteria might make a collagenase, breaks down collagen. Some might make it a polymelendase, breaks down hyaluronic acid. Different ways to get in between your cells or through your cells to invade. Ability to bypass or overcome host defense mechanisms. We'll talk about that a lot as we go through. But maybe something like we've already talked about, like a capsule. Well, maybe that makes a bacterial cell so big that the macrophage can't engulf it. Or maybe the macrophage takes it up and the capsule protects it from being broken down. Or it makes some kind of virulence factor where it hangs out in the phagosome but prevents the phagosome and the lysosome from fusing. I remember that we talked about that in immunology. I've been fed immunology. Oh, not that. Okay. I don't know if we talked about that here yet. So if you have a macrophage that say engulfs a bacterial cell, it's going to be taken into what? What's it? Phagosome is what it's going to be contained in. You also have inside macrophages, you have lysosomes. Lysosomes are where all the degradative enzymes are that ultimately are used to break down the bacterial cell. So there's a process in there with a phagosome that has a bacterial cell in it, and a lysosome that has all the things it's supposed to break down the bacterial cell merge together to make a lysosome. There's some bacteria that secrete things that prevent that fusion from happening. So I mean, that's a great environment for bacterial cell to hang out in as a phagosome because there's nothing in there that's going to harm it. It's hanging out inside of one of your white blood cells, so your immune system's not going to get rid of it because it's not going to recognize your white blood cells as foreign. There's some organisms that do that. There's some microorganisms that escape that phagosome and just hang out inside the body, which is really nice environment for them too. Nothing there going to be in. So we'll talk about some of those examples here in a little bit. Pathogenesis, that's a real hard one. Ability to make toxins. So not too hard to understand there. Some different kinds of toxins. You can make exotoxins, so bacterial cells you can make exotoxins, which are exactly what it sounds like. The toxins are secreted. So leukocytes are going to kill white blood cells. That's an example of an exotoxin. Endotoxin is only one example. Maybe tell me what an endotoxin is. What's an endotoxin?
So it's inside the body of the
No, more specific. What's the competitor to bacterial cell that's endotoxin?
Where do they have
Yeah, so you have a little bit of polysaccharide or LPS. And what's the competitor to LPS that's the endotoxin part? Lipid, right? What bacterial cells make lipopolysaccharide?
Negative.
Gram negatives, right? All gram negatives are made lipopolysaccharide. So this is kind of the example I've given you a few times in here. Usually the typical kind of scenario you see with this is you have somebody that's elderly, they're in a nursing home, they have a urinary catheter, they get a UTI, and no one notices that they have a UTI. That progresses into urosepsis where it gets into the bloodstream, the bacterium, usually it's E. coli. We'll come back to that a little bit later as well. E. coli gets in the bloodstream, and then by the time they actually go to seek treatment, they have a ton of E. coli in their bloodstream, and if you treat that with an antibiotic, it's going to release that lipid A. different virulence factors. Adhesins promote colonization through adherence, that allow adherence to cells in your body. There's different kinds of adhesins you can have. Some adhesins are going to be on the surface of the bacterial cell, like right on the surface of the bacterial cell. Other adhesins are going to be on the ends of villi. They're called tip adhesins. Again, sometimes we're not super original with our naming, which is good because it actually tells you what it does. You see that on the end of villi sometimes, tip adhesins. Now, antiphagocytic surface proteins and capsules, we'll talk a little bit about that. IgG, antibody binding surface proteins, we'll come back to that and talk about this in a little bit. But essentially, there are some microorganisms like Staphylococcus aureus can make a protein A. And what the protein A does, if you have an antibody, say you have this antibody, which region's the, let's do this. Which region's the constant region, A or B? B, right? So B is your constant region. Which one's your, well, I need to figure out how to spell constant today. Which one's your variable region? A, right? So this is where things that are foreign are supposed to be recognized through this, right? And this is recognized by some white blood cells that have a constant region receptor called an FC receptor. So what happens is you get this protein A that binds here and blocks this interaction. So Staph aureus can make a protein A. Some streptococcus species can make a protein called protein G. It does exactly the same thing. It binds on this constant region, so you don't have that interaction between the antibody and some white blood cells. So that's one way that bacterial cells can get around different antibody-mediated immune responses to binding on that constant region, blocking the interaction with white blood cells. Endotoxin we talked about. Antigenic switching we have not talked about a lot. There's two examples of that. One of them is called phase variation. These terms kind of get switched around back and forth. Phase variation means that the microorganism is going to change what it's expressing. So it's either going to be making it or it's not going to be making it. So phase variation is more of an on or an off. So an example of that would be like salmonella. Salmonella can make a couple different kinds of flagella. The flagellans are the proteins that make up the flagella. So, so phase variation is going to be on or off. Again, a lot of times these two get kind of flipped back and forth. But phase variation is supposed to be on or off. So an example of this would be like salmonella. Salmonella can make a couple different kinds of flagellants. It can make one, you don't even know these, it's just to give you an example. It's called FLIC, another one's called FLJB. So depending on the condition it's in, it can make a flagellum or it can make flagella that are made of this. Or it turns off the expression of this and it can make flagella that are made of that. So it's on or an off. Antigenic variation, there's a lot more examples of that. Antigenic variation is where it's going to change the relative ratio of the different kinds of, say, proteins it makes on its surface. One example of that is Neisseria meningitis, we're falling to pieces here. Neisseria meningitidis, which can cause meningitis, and Neisseria gonorrhoeae, which causes gonorrhea. And what it can do is it can change the relative expression of, usually it's villi on its surface. So it can make a few different kinds of villi. And what it does is say, it starts out making a bunch of these
giving you a random letter, like pili-A, it makes a bunch of that, and then it starts to decrease the expression of pili-A. It still might make a little bit of it, but it makes a whole buttload of pili-B. And then after a little bit longer infection, it starts to down express the pili-B. So it's still making a little bit A and B, but it starts making C. So it's not really turning on or off the expression, it's just changing the amount of these different things that you're making. So that's classical antigenic variation. So it's changing the antigen on its surface. Why does that matter? How's that going to help it? Think about how your immune system responds to things. Yeah, it doesn't get recognized. So your immune response starts to make antibodies, and you've seen how that works. We talked about that here a couple lectures ago, a couple days ago. Your body starts making antibodies to pili-A, and then as soon as you start to get a good antibody response to pili-A, it downregulates the expression of pili-A and starts making pili-B. So your immune response can't keep up with it, it just keeps switching. There's a bunch of examples of different bacteria that do that, and parasites too.
What's the difference between antigenic and phase variation?
So again, phase variation is going to be completely on or completely off. So like for the salmonella example, and that's one of the few examples I can give you, like a just straight up phase variation, because most of the examples are going to be antigenic variation. Phase variation means it's on or off. So salmonella is either going to make that FLIC flagella, or it's going to make FLJB. It completely turns one off and turns the other one on. Antigenic variation, it's not going to really turn anything off, it's just going to decrease expression and increase expression of something else. So it's not as absolute, I guess you could say. Invasons, this is what it sounds like. Invasons are going to allow the bacterial cell to invade. Invade human cells are not normally phagocytic, escape immune response. Some of these things as we go through too. Growth properties and capacity for intracellular growth. Some bacteria have the ability to grow inside your cells. If they're growing inside your cells, your immune system's not going to recognize it because it's inside your cells. Ability to form biofilms. Usually the easy example to understand of this is somebody has like a knee transplant or something. You have Staphylococcus epidermidis on your skin. They put in a new knee, and some of that staph epi gets on that knee replacement, and then it makes a biofilm. Once it makes a biofilm, bacteria becomes super resistant to antibiotics, so there's no real good way to treat that. You have to take out the knee. You have to go through another surgery, take out the knee, put it in a joint space that has antimicrobials in it, treat it with antibiotics, go back in, take out the space, or put in a new knee. Not good. That happens. It's almost impossible for it not to happen every once in a while because you carry all this stuff on outside your body, and it finds its way in sometimes. Do I talk about that? Maybe not. Let's talk about it for a little bit. So when you talk about biofilms, a lot of times you will see very odd presentations in humans. So you'll see like these odd, like fungal biofilms on the heart valve and things like that. Or you find these odd disseminations of things all throughout the body that you normally wouldn't suspect to go systemic as fast as they do. What you see a lot of anymore is there is a lot of like IV drug use, heroin, fentanyl, whatever. Drug dealers don't have great QC, so you end up with things inside the drugs that are environmental, aspergilluses, and I'll give you an example. This happened up in Greensville around when SARS-CoV-2 started to be a thing. There was a heroin dealer. There's a guy dealing heroin, and all the people that had the heroin drug dealer. All the patients that came in had hepatitis. Because somehow they got, I think it was Hep A. Actually, I know it's Hep A. Sorry, I'm trying to recall. It's been a while. Yeah, it was, I think it was meth, and they had Hep A. Yes. So they had this Hep A epidemic, but at the same time, they also started having the SARS-CoV-2 epidemic. The SARS-CoV-2 epidemic kind of took precedence over the Hep A, and everybody started watching the SARS-CoV-2. But in the background, it's like Hep A, Hep A, Hep A, Hep A, Hep A was going up, and nobody was paying much attention to it. But that's that. So again, not good quality control. Somehow that drug dealer got poop-tainted methamphetamine, and then all the people who took the meth had meth. Where this came into play with the general populace in that area is, for whatever reason, just coincidentally probably, a lot of the people that were being supplied with that poop-tainted methamphetamine worked in kitchens at restaurants around the area. So they would take methamphetamine, they would get infected with Hep A, they would go to the bathroom, they wouldn't wash their hands, they'd go make their food. I'm like, literally everybody had Hep A, whether you took methamphetamine or not. So,
Where was this at?
here you go. That one kind of got underneath the radar a little bit. I was like, wow, I can hear something. I was like, ooh, that's a big number. And they're like, you don't know what's going on. I'm like, you know what's going on. I don't know if they ever got the guy or not. I would assume they did. It stopped, so I mean, they had to try. But, yeah. Point being, because I'm really often have support that one, these people don't really give a crap whether you get poop tainted or whatever tainted drugs. They're drug dealers, they don't care. What you see in people that do IV drugs sometimes is they'll end up with these heart conditions from different pathogens that get tainted inside the drugs, and then they'll have all these things like fungal biofilms on their heart valves and stuff. That's really weird. You don't see that. You start to see a lot more of that now because there's a lot more of that kind of drug use, but I mean 20 years ago that wasn't a thing. Somebody came in with some of the things now you see on like heart valves, and you'd be like, what? Like it was real rare. Now you start to see things like that. It's like, oh, probably. I had a case a while back. I actually never written up yet, where there was an immunocompetent person, and she really wasn't anything wrong with her, immuno-wise. She liked to use like a vape pen. I don't smoke. But to my understanding, you can take like the little pictures out of the vape pens, and you can put things into those. And she injected with like a little bit of something like THC or something into the vape pen. And when she did that, there's aspergillus literally like everywhere. And she had the worst case of like aspergillus ever. I mean, like so bad, like I know an infectious disease doctor that was an IV doctor in the military for years. He's been doing this by 40 years, and when they actually went down into the lungs with a scope, they were going down through it. There was this big aspergillus fungal balls, and if they bumped them, they would detach and like kind of levitate in the respiratory tract as they went down with the scope. You don't see that kind of stuff unless somebody's ingesting or somehow taking in things that you don't really take in. Like I remember what he told me was, he's like, I've seen people with like late stage like AIDS and literally don't have like an immune system. And he's like, and these infections in them isn't this bad. It's just essentially she nebulized aspergillus into herself, like trying to, like I said, you go into practice, if you go to med school or wherever, you're going to see a lot of weird presentations from things that like normally you wouldn't see anymore. Congenital syphilis is on the rise. That hasn't been a thing for 40 years. And now you're seeing more cases of that. People with vaccine hesitancy that don't want to take some of the vaccines. I hope to God y'all don't see polio, but I'm starting to kind of wonder, y'all might see some polio. What's the other one? Measles. Measles is a big one where we're starting to see that. Problem is, if you go into like different positions, like say you go to a family medicine doctor and you have measles, a family medicine doctor has never saw measles in their life. The only place they've ever seen measles in is actually because measles hasn't existed. So I don't know about y'all, but I really don't want somebody to treat me that's literally never seen anything besides a picture in a textbook from 1976. So some of it gets a little scary. Okay. Extracellular gene products, we talked about some of these, things like collagenase to break down collagen, hyaluronidase to break down hyaluronic acid. These are the things, these are like the glue that holds your cells together. So if you can figure out a way to get around those in your microorganism, then you can invade. IgA, antibody degraded protease. Does anybody know where you find IgA at? In secretions. So like in like your respiratory tract, in breast milk, things like that. So if you have, say, you're in your respiratory tract and you have that kind of mucousy layer that coats your respiratory tract, inside that, not only does the bacterial cells, they have to get through that mucus layer, but they have to get through that mucus layer that has IgA. So some of the bacteria make proteases to break down antibodies. Yeah. IgG is the most in the blood. IgA is the most, that's where it gets kind of confusing. So if you're looking at the body in totality, blood, organs, secretions, everything, IgA is the most. If you're only looking at which one's the most in the bloodstream, it's IgG. That makes sense? I know that sounds word kind of confusing sometimes. Yeah, IgA is the most overall, IgG is the most in the blood. Yeah, most people talk about it, they're talking about the blood. IgG is the one in the bloodstream. Exotoxins we talked about, protein secretion systems. Don't really need to worry too much about that, but essentially what that means is there's different mechanisms, protein secretion systems, that these bacteria can use to actually get the protein virulence factors out of the bacterial cell. That's all I was talking about. There's different secretion systems for things like siderophores, which are going to be used to gather iron, and all of those secretion systems are going to be controlled by gene expression based on the environment they're in. Different bacteria colonize different parts of the body depending on what receptors they recognize in different parts of the body. Sometimes this is closest to the portal of entry, so wherever you take this microorganism in at. Again, colonization is going to require a bunch of different bacterial virulence factors. You're going to allow that bacteria to remain at the site, so that's essentially saying allow the bacterial cell to attach to you somehow, survive things in your body after they attach. So if it's on a mucosal surface, a way to get around the mucus, a way to get around the IgA, depending on where it's at, maybe a way to get around the lysozyme. So that's a survival, and ways to get around the immune response too. And then also, it is a living organism, so they have to figure out a way to actually obtain food too. So some way to live. One of the ways this happens with a lot of respiratory tract pathogens is they can actually use the mucus, and actually use it as herbicides. So they'll use it as food. So the great reason to use that instead of like glucose or something. Bacteria adhere to these different cell structures, like in the bladder, intestine, blood vessels, and then they can't be washed away. So we talked about that too. So again, the main thing is they have to attach somehow. This is a bunch of different ways to attach. No, you don't have to know all these examples. Just kind of know what the different structures are. You don't have to know all these different examples. We'll talk about a lot of these as we go through. Villi and fimbriae, remember we talked about villi. Generally, villi is going to mean they're longer proteinaceous structures that emanate from the surface of the bacterial cell. There's not a ton of them, usually between like 10 and 20. Fimbriae are short, and there's a ton of them. A lot of gram-negatives use this for attachment, villi and fimbriae. Capsules are used for a bunch of different things, which we already talked about. There's a bunch of different microorganisms that make capsules. There's a couple examples there, and we'll talk more about those. Here we're talking about respiratory tract pathogens over the next month or so. Techoic acid, a lot of gram positives are going to use techoic acid. Techoic acid, remember if I told you if you see anything with tech in it, it's going to be a gram positive. Techoic acid not only kind of stabilizes the cell surface of these gram positive microorganisms, but the techoic acid can actually be used for adherence to cell surfaces as well. Some types of E. coli, which we wouldn't talk about this until the spring, make something called colonizing factors, which allow it to adhere to intestinal epithelial cells. We'll talk more about, there's a lot of different kinds of E. coli, ETAC and EPEC and EHAC and EIEIO, I feel like sometimes. There's a bunch of them. We talk about that more when we talk about GI tract pathogens and infectious diseases, but there's about 6 of them. 7 if you include the ones that cause UTIs. IgA proteases, we talked about that, some different examples of microorganisms that make IgA proteases. What I do want you to kind of get from this, even though we haven't really talked about it yet, Neisseria meningitidis. Anytime you see SPP, it just means species. But I'll give you meningitis as an example. Neisseria meningitidis, Haemophilus influenzae, Streptococcus pneumoniae. Those are things that can affect your respiratory tract. As a respiratory tract infection, they have a propensity of finding their way out of your respiratory tract to your blood-brain barrier, causing inflammation there, and ending up in your cerebrospinal fluid, and they end up causing meningitis. How do they do that? Well, they have to initially establish infection in the respiratory tract, and one of the ways they do that is they make IgA proteases so they can adhere and colonize your respiratory tract. And if that infection is allowed to get bad enough, oftentimes it finds its way to your central nervous system. Because of really, really nasty meningitis. So they all make IgA proteases. Biofilms, a bunch of bacteria that make biofilms, there are some examples there. We talk about different things that biofilms can contain the bacteria do. So keep them hydrated, allow it to attach to surfaces, evade immune responses, etc., etc., etc. Capsules are important for lots of things. Again, certain bacterial cells are going to make villi, sometimes they're referred to as fimbriae. Again, those terms are used interchangeably anymore. And a lot of them are going to make capsules as well. And that those structures are going to allow adherence to different surfaces in your body. A lot of times when we talk about adhesins, the targets on your cells are going to be sugar-based, they're going to be carbohydrate-based. Sugar binding activity defines the proteins as lectins, so means they refer to these adhesins as lectins. Some bacteria have these adhesins on the surface, some of them have the tip adhesins on the fimbriae or villi. This depends on the microorganism. Please fill up what I do not know. That's my job. So if you look at these different microorganisms, again, they're going to have different adhesins. This is a very short list of a very long list of adhesins. I highlighted some of the more important ones in red that we'll talk about as we go throughout the semester at least. You do not need to know all these until we talk about specifically the different types of infections. But like Staph aureus makes a clumping factor that recognizes fibrinogen, fibronectin. Different Staphylococcus species can make these things called M strands. I'm not going to read all these. Neisseria gonorrhoeae makes fimbriae. Remember I talked about Neisseria, they can undergo antigenic variation, so we can antigenically vary these fimbriae. Treponema pallidum causes syphilis. It makes different proteins, P1 through P3. It can also undergo somewhat a little bit of antigenic variation between these different P proteins. We won't talk about a lot of these in here necessarily, but just to know there's different ones. Yeah.
So like actually nothing.
Just the ones that are in red. And at this point, not even really those yet, because we'll talk about those specifically as we go throughout, like later on, when we specifically talk about these different bacteria, we might talk about those. Like E. coli, fimbriae, that's the established urinary tract infection. Vibrio cholerae makes type 4 villi, that's how to establish cholera. so we'll probably talk about. So you call in mixed type 1 fibrin, P-fibrin, fibrin, mixed type 4 fibrin. Again, biofilms are going to aid in attachment. They're also going to confer resistance to things like antibiotics, and also they're going to confer resistance to things like your immune response. You'll see these on surgical implants, valves, prosthetic joints, inguinal catheters, things that the biofilm can establish themselves on. A bacteria bound with sticky web polysaccharides, proteins, and DNA that bind cells together into this surface. We talked about form sensing a little bit, but when these bacterial cells are in close proximity to one another, they can release small chemical mediators referred to as form sensing molecules. An example of that is something like homoserine lactone, one of our classical ones. Essentially, you have bacterial cells that they all make a set amount of homoserine lactone. They can also sense the relative amount of homoserine lactone in the environment. The higher that concentration of homoserine lactone, the more bacterial cells that the bacterial cells know are surrounding them in that population. So they can use that to control their growth. So if they are growing too much, they might use up all their food source. So if the homoserine lactone goes too high, they slow down their growth. So they don't essentially start themselves up. That's an example. You have diabetic patients sometimes that have chronic infections that they have a hard time actually getting healed. A lot of times that's because those microorganisms cause infection in the biofilm. A real good example of this is cystic fibrosis patients. Oftentimes you'll see people that have cystic fibrosis. We always talk about cystic fibrosis in the classes, right? It's bad for all around. The cystic fibrosis patients have a mutation in CFTR, ductance factor, transmembrane regulator, or something like that. Essentially what happens is their lungs make a bunch of these really like mucously thick secretions the normal lung would not make. What happens is these people end up getting pseudomonas in their lungs because of pseudomonas infection, and the pseudomonas makes these really thick biofilms in cystic fibrosis patients' lungs. They also change the way they behave. Like normally, if you grew pseudomonas here in the lab or whatever, it respires aerobically, which means it needs oxygen for metabolism. It's motile, so it can move. In a cystic fibrosis patient's lungs, it becomes sessile, which means it doesn't move anymore. It starts making this substance called alginate. It can undergo more of an anaerobic respiration. It doesn't even, like, you compare bacteria like Pseudomonas to the CFH1 and Pseudomonas like a honey tradition here, it doesn't even look like the same bacteria. Like, they behave completely differently based on the environment they're in. But once it's in the lung, it starts to make these biofilms, and you can't eradicate the infection. So you can give these people antibiotics, but it's fluid because really the only thing you do is just kind of suppress the amount of bacterial cells there, the amount of Pseudomonas cells there, but it typically doesn't actually get rid of all these cells. I mean, you're talking about like hardcore antibiotics where you're like nebulizing and inhaling antibiotics like tobramycin and things like that. And oftentimes it still doesn't kill all the Pseudomonas. You just keep on treating with antibiotics and you can at least control the amount of bacterial cells that are there. But you generally don't get rid of it. And that's because they make that biofilm that makes it really resistant. And then those cause really nasty infections in CF patients. CF patients have a lot of problems with respiratory tract infections, particularly Pseudomonas, that's similar back and things like that. This is the process by which a biofilm films. I can walk through this a little bit with you. You have a substrate. Bacterial cells start to make different molecules that allow that surface to be ready for adherence. The cells, the bacterial cells adhere. Some of them will stay adhered and they will start to make a biofilm. Some of them can break off from that biofilm and go form a new biofilm somewhere else. This is why you really worry about things if you have somebody that has like a heart valve transplant or something like that and there's biofilm on it. That's in your bloodstream. So if there's a biofilm on that, and a little piece of that biofilm breaks off, it can get in your bloodstream and then establish a biofilm somewhere else too. So it can cause a really nasty systemic infection. What biofilm looks like on your teeth, you can see it with your eyes and stain or with a microscope. This is why you go to your teeth cleaned about every 6 months to get off the plaque. Plaque is the formation of biofilms from those microorganisms in your oral cavity, specifically on your teeth. If you don't go do that, you can get periodontal disease where it gets deeper into the gums and into the underlying bone as well. There are some different bacteria in the mouth that can cause some really nasty infections. Some people have an organism called Aggregatibacter. Aggregatibacter can cause a really nasty periodontal disease where it gets into the underlying bone. There's different streptococcus species you have in your mouth that can cause a focal infection. Everyone remember what focal infection is? What's a focal infection? How's a focal infection spread?
An area that's initially confined to a local area that's become somewhere else.
Yeah, so usually it's going to go through the tissues as opposed to something that's like a bloodborne pathogen, like sepsis. So usually a focal infection is going to migrate through the tissue from one area to an adjacent area. I actually gave you an example of this today, and I didn't even tell you it was a focal infection. Talk about like the serious meningitis goes through your respiratory tract to your central nervous system. Well, those aren't far apart. So that's how it gets there. It's a focal infection, it's not systemic. Since we're talking about the oral cavity, you have something like a streptococcus species, usually it's virulent streptococcus, we'll talk about that a little bit later as well. It gets out of the mouth, travels through the tissue, and ends up in your heart. It can cause a really nasty endocarditis. There's a lot of things in your mouth that they get out of your mouth can cause some really nasty infections. Another one that we might talk about in here, there's a paper in Nature a while back where they were looking at a bacterium called Porphyromonas gingivalis, I think, a long name. Porphyromonas gingivalis can make gingipans, which are protein, and they have found those proteins in the brains of Alzheimer's patients. All Alzheimer's patients they look at, they find this protein. So somehow that microorganism, or at least the proteins it makes, gets out of your oral cavity into your brain, and it's somehow associated with Alzheimer's disease. How that is, I don't know. But yeah, a lot of nasties, usually, if given the opportunity to escape that. Different ways to overcome host defenses. We talked about this a little bit, but I'll give you some examples here. Inhibition of phagocytic engulfment. So the best way to prohibit getting broken up by a macrophage is to not get engulfed by the macrophage in the first place. Survival inside the macrophage, so they can inhibit this phagosome-lysosome fusion, they can survive inside the phagolysosome, or they can escape from that phagosome and live inside the cytoplasm. Some all examples we kind of talked about already. Or they can just straight up just kill the white blood cell. Just kill it. They can do that before they get ingested in the first place, or once they get ingested, they can kill it from the inside. Different examples of different microorganisms do these things. What this kind of looks like. Again, bacteria get taken in the phagosome and the lysosome fuse to make a phagolysosome. And in theory, what's supposed to happen is the bacterial cell gets broken down, and then those pieces of parts get presented on the cell surface. And the rest of it just gets exocytosis. Capsules we've talked about. So we don't need to really talk about them again. Different things you see here that allow all these different bacteria to avoid being destroyed. And a nice little, holy God, what's the word I'm looking for? Mnemonic, thank you. Wow. Ooh, yeah. Someday I'll be a wicked idiot. So if you look down here at the bottom, some killers have pretty nice capsules. That's a nice way to remember some of these microorganisms that really rely on a capsule to cause a really nasty infection and evade their immune response. Streptococcus pneumoniae, Klebsiella pneumoniae, Haemophilus influenzae, Pseudomonas aeruginosus, Neisseria meningitis, and Cryptococcus neoformis, which Cryptococcus we won't talk about in here. But you see that, it's not bacterial, but you see that in AIDS patients a lot of times. They can get a very lethal infection, but secondary to HIV infection. Any capsule is going to be antiphagocytic, so like these ones. Saline layers can also be antiphagocytic. Pseudomonas can not only make a capsule, can also make a saline layer depending on the environment it's in. Have you ever what's the difference between a saline layer and a capsule? Any difference?
Slime would be like washed off.
Saline layer is not as heavily attached to the underlying material, so capsule is it's attached. It's not going. M proteins we haven't really talked about a lot in here yet. Sometimes you see M referred to mimicry. So those M proteins, there's different things those M proteins can do. Group H streptococci make these M proteins. They can have structures similar to some of the proteins in your body. So the M sometimes stands for mimicry, molecular mimicry. So your immune system might not recognize these group A strep because they express these M proteins and essentially make the streptococci look like your cells to some extent. So your immune response doesn't protect them. That can also help them evade being phagocytosed because they're not going to be recognized as foreign. In E. coli can make O or K antigens. We'll talk about that in spring. Salmonella typhi can make VI antigen. That's not antigen 6. That stands for virulence, VI. That's antiphagocytic for Salmonella. Protein A, we already talked about. Staph aureus makes that. Binds on constellation of antibodies. And a bunch of them can make villi. Neisseria gonorrhoeae is one of them. Remember, Neisseria gonorrhoeae is one of the ones that can undergo antigenic variation with this villi. So just which villi is making the relative ratios of villi is making this a immune response. We talked about this as well. So remember I said some bacteria will actually hang out in the phagosome and they'll prevent the phagosome and the lysosome from fusing with one another. So they can just hang out in the phagosome and not be destroyed. These are some examples of bacteria that do that. Salmonella, different species of salmonella can do that. We won't talk about salmonella here. We talked about that infectious diseases in the spring. But essentially salmonella, what we found over the years are all the species of salmonella were so closely related to one another that they weren't actually different enough from one another to be classified as different species. So pretty much everything is called Salmonella enterica, and then they're categorized in different serobars. So everything is Salmonella enterica, serobar, typhimurium, color Suez, whatever. There's a bunch of different ones. Really the only one that remains that causes human pathogenesis that's not under Salmonella enterica is Bongorai. Wouldn't be surprised if they throw it in there and mention someday too. But most of the time we're talking about enterica. Mycobacterium tuberculosis does this. Specifically, I'm doing lung macrophages. Legionella pneumophila. We'll talk about that a little bit. We talk about respiratory tract pathogens that causes Legionnaire's disease and different chlamydia species. These are ones that survive inside the phagolysosome. Bacillus anthracis. Bacillus anthracis is kind of an oddball. So capsules, remember, are usually made out of polysaccharide. Bacillus anthracis is made out of peptide. So it's more protein based. Mycobacterium tuberculosis, Staphylococcus aureus. These ones are going to survive inside the phagolysosome. So they all possess mechanisms to survive intracellular. So the macrophage doesn't kill them. So they're hanging out inside the macrophage. They have ways to get around these degradative enzymes in the lysosome. They can hang out inside the macrophage, and then they go undetected by your immune response. Or they can escape from the phagosome. This is when it starts to get a little interesting. So there are some microorganisms they can escape before the phagosome fuses with the lysosome. Rickettsia is an example of this type of rickettsial species. Talk about that in spring as well. I feel like I say that a lot. Produce a phospholipase enzyme that lyses the phagosome membrane within about 30 seconds of ingestion. So it takes it in, and then it makes a phospholipase that breaks it down and it hangs out inside of it. Listeria does this. Listeria is a GI pathogen. Legionella pneumophila, again, causes Legionnaire's disease. And Francisella tularensis, we'll talk about a little bit when we talk about respiratory tract pathogens. Francisella tularensis causes tularemia. Sometimes you hear that referred to as rabbit fever. We'll come back to that here in a few weeks. Some of these microorganisms can do some very weird things. This is an example of what Listeria can do. Listeria can make a couple different things called internalins. That should say INTA and INTB. The textbook I took this out of apparently had a typographical error on that. These are internalins. They aid the bacterial cell in being internalized. That's all that's saying. So they're taken inside, they're taken into this vacuole, and then they make a lipase. Lipase will allow this vacuole to be degraded, and then you have the bacterial cell out into the cytoplasm. For a lot of microorganisms, that's kind of enough. And inside one of your host cells, if I remember correctly, I think this is an endothelial cell somewhere. But they can hang out inside the cytoplasm. Cytoplasm is nutrient rich and be protected from your immune response. A lot of microorganisms are cool with that. Listeria is not cool with that. It wants to spread to other neighboring cells. So what it does is it makes this protein called ACT-A. And what ACT-A aids in doing is robbing your cells of actin. It polymerizes those actin behind the bacterial cell and makes something referred to as an actin rocket, and uses the polymerization of those active proteins to literally shoot from one cell through that cell membrane and the cell membrane of the neighboring cell to gain entry into that neighboring cell and spread. There's not a lot of bacteria that can do this. Listeria is one of them. So what happens is it shoots over this neighboring cell, but then it has this kind of two-layer cell membrane surrounding it. Cell membrane from this first cell infected, and the cell membrane that literally was shot through. So it's got two-layer membrane. It makes a bunch of other phospholipases. Same lipase made over here, this LLL. It also makes a couple phospholipase C's, phospholipase CA and phospholipase CA, excuse me, to degrade all the cell membrane and then become released out into the cytoplasm again. And then it'll make ACT-A again, use ACT-A to shoot over to the next cell, over and over and over and over and over again. Again, the ways in which they've managed to figure out ways to invade and escape immune responses have become a bit complex for some of these. Legionella pneumophila goes about this in a different way. It makes different virulence factors that induce something called coiling phagocytosis, which looks exactly like that, where the pseudopod is actually induced to kind of coil around the bacterial cell, ends up bringing the bacterial cell in. But ultimately what ends up happening is it gets put in this phagosome that's studded with ribosomes. So to the inside of your cell, it looks like rough endoplasmic reticulum. So it looks rough ER. And it just hangs out in that rough ER-like structure. Eventually, that structure will lyse and there's enough bacterial cells that grows inside of it. Bacterial cells become released into the cytoplasm and they can escape the cytoplasm. So that's how Legionnaire's disease works. Some bacteria, we talked about this a little bit too, they don't even mess around with the white blood cells. They just kill the white blood cell before it's ever taken up. And a lot of gram-positives do this. They secrete enzymes called hemolysin. The hemolysins usually are, they're usually pores. Essentially, they just poke pores inside these in the red blood cells. Streptococci, streptolysin is an example of a virulence factor associated with phagocytes and forced phagocytosis. leukocyte, methylstatic toxide, exotoxin A is made with pseudomonas, Bordetella pertussis, which comes with pertussis, whooping cough, makes an adenylate cyclase that does this, different ways to kill the phagocytes before we were taken up in the first place. And then after ingestion, so they're already taken up, they go from the inside, they release different toxins while inside the cells, like a macrophage. Examples of microorganisms that do this, Mycobacterium, Brucella, Listeria, they can do it from inside the phagocytic cells. Different ways to evade complement, hide LPS peptide glycan, so they can't be recognized through the complement pathways. So essentially, things that are on the outside of the cell, on the outside of the bacterial cell, that might be recognized by something like toll-like receptor, they just shield all those proteins with cancer. The Cerebrinagitis, Haemophilus influenzae, they can attach sialic acid residues in their surface. What that does, you make a lot of sialic acid on your cell surfaces as well. Essentially, the bacterial cells start to look like your cells, and they evade the immune response that way. Pseudomonas can make elastase, which inactivates different components of complement. Streptococcus pyogenes, which is group A strep, pretty much the main member of group A strep is pyogenes. Makes an M protein that can destroy different components of complement pathway. One of the things I want you to get from that is look at what it's targeting. So remember I told you I didn't want you to know the components of the complement pathways as the breakpoints. So it's targeting things that are the breakpoints of the pathways that are going to shut down a lot of things at the same time. This is when things like that become more important. It doesn't necessarily matter the individual steps of all those different complement pathways. What matters is this microorganism is targeting something that's a breakpoint for pretty much all of those pathways. So essentially it just shuts down complement. We talked about this. Staphylococcus makes protein A, streptococcus makes protein G, binds onto the constant region of the antibody so it can't recognize constant region receptors on